All right. Yeah. So thank you very much. I'm very grateful to Mike and to the other organizers for inviting me at this very beautiful conference in this really amazing venue. Okay, it's been really like a good learning experience and a pleasure so far. And I would like to start with this picture that has been shown already a few times, a few times during this meeting, which is one of the most famous illustration about turbulence. Okay, and it's really a remarkable picture by Leonardo da Vinci, also because it's not a typical illustration in the sense that it's not a drawing or something that you can see. If you pour fluid from above to create a turbulent flow, definitely don't see this much structure, okay? So this is an illustration of a theory, okay, of a theoretical idea. This is how Leonardo envisioned turbulence. And it's a beautiful multi-scale structure that consists of features, eddies, vortices, at several different length scales. And what that makes it really incredible is that, as you know, presumably even better than me, this is in essence the very same idea that Richardson put forward a century later to explain the nature of the energy cascade. Okay, big worlds have little worlds that feed on their velocity, and little worlds have lesser worlds, and so on to viscosity. Okay, so this is a little Swiftian poem by which Richardson explained that turbulence is ultimately the trick that nature has devised in order to dissipate energy when dissipation is negligible. So for this reason, it should be rather surprising that turbulence somehow can be observed also in biological active fluids. Okay, so these are fluids made of biological constituents in which the velocity is of the order of 1 micron per second. Okay, then almost everything in biology goes at the speed of 1 micron per second. Okay, so in this regime, dissipation dominates over inertia. Nevertheless, if you look at these videos, this is, for instance, is an example of an active pneumatic fluid made of microtubules and kinesin. It was pioneered by my good friend Zvonimir Dojic in 2 dimensions. And I will explain what that is in a second, but Mike already has done a wonderful job in introducing the main ingredients here. Well, so this flow consists of several vortices, jets, swirls, and so on. So it is very much reminiscent of a turbulent fluid in 2D, even though here Reynolds number is essentially 0. In the second video here, instead, what I have is sperm cells. Okay, this is a work that came out some time ago. And once again, you keep seeing the same swirling motion. And finally, the last piece of work, which is much more recent, these are human bronchial epithelial cells. So those are cells that are plated on a 2-dimensional substrate. It's a different kind of turbulence. It's a decaying kind of turbulence. As you can see, the cells eventually come to a rest, and yet they do display similar patterns. So if you believe, if you have the impression that the swirling motion in these 3 videos is visually similar, you are very much on the right track. In fact, it's not just visually similar, but it's statistically identical, in the sense that all the metrics that we can extract, the statistical metrics that we can extract from these experiments actually give us the same results. Now, there are several reasons to be excited, I believe, but I'd like to highlight 2 of them. One is universality. So this is one of the key concepts in this meeting, right? So universality is a crucial idea in statistical physics. Unfortunately, when you get to deal with biological systems, you soon realize that perhaps this idea is too ambitious for biology, okay, especially for cells, because cells are really complicated, okay? There are a lot of different effects that might or might not come into play, depending, for instance, on the environmental conditions. So this is something that biologists called phenotypical plasticity, has nothing to do with material plasticity, and yet is a major hurdle if you try to model biological systems. Nevertheless, in these videos, it seems that there is a notion of universality in the traditional sense, okay? Exponents that are the same and do not depend on the material parameters. The second reason to be excited, which this one is very close to my heart, is that we have a hydrodynamic theory that can actually account for that. okay, that can describe this type of motion. Now, there are people in the community who have been advocating this idea for at least 1 decade, possibly more. So perhaps I should not be surprised, but I'm a bit naive, so I am surprised that you can actually describe the collective motion of cells using differential equations, okay? So all the systems that I showed so far belong in the broad category of active matter, okay, active systems. I will not going to review this again because Mike has already done a wonderful job. I will just point out that the idea of active matter in physics, at least in the physics community, started almost 25 years ago. And really the main idea was to describe motion at the macroscopic scale, collective motion at the macroscopic scale, like in animals and so on, bird, fish, etc. But that soon after, somehow the same ideas somehow managed to spread across all these landscapes. Okay, and now one of the most recent topics in the physics of active matter is really these eukaryotic cells, tissues, cell layers, which I believe are really, really interesting systems. And I'm sure that we will see like a blossoming of physics of eukaryotic cells. I also want to advertise that in 2020 it's the 25th anniversary of active matter, so there will be like some events around the world. One will be in Santa Barbara. And one will be at Leiden in the Lawrence Center. So if you want to celebrate with us, keep a look. Now, the type of systems that I showed a few slides ago are a subset of active systems, which we call active nematic liquid crystals, okay? So those, I will explain what they are in a second, but they are liquid crystals of biological origin that can be assembled in different ways from various biological constituents, cytoskeletal filaments, for instance, microtubules, acting bacteria, or also eukaryotic cells. Here is my one slide introduction to liquid crystal for the people who are not familiar with this topic. Liquid crystals are fluids which are orientationally ordered. They are made of molecules which are elongated. These are two examples of molecules which are commonly used in studies on liquid crystals. 5Cb is literally the one that is inside your iPhone. And if you manage to zoom on a little volume element here, what you would see is that all these molecules are roughly aligned with one another, okay? Now, these molecules have a polarity in the sense that the head and the tail are clearly different from one another. Nevertheless, when they form these dense phases, they are insensitive to this polarity. So you can really think of it as a fluid made of rods, okay, which are head-tail symmetric, and they pack accordingly to their orientation. Such a mean orientation is called the director field, and is typically indicated with the letter N. Okay, I want to avoid jargon, so I'm introducing my jargon. Now, active nematic liquid crystals are somehow similar. So in this case, also if you do these little experiments, if you imagine to zoom on a little volume element, what you will see is, again, these rod-like molecules which are oriented with one another. The difference now is that these molecules are able to move. And as a consequence of this motion, these molecules will exert a stress on the boundary of a volume element. Okay, and this stress is clearly oriented as the director field, as the average orientation of these molecules. Now, this stress vector, okay, can be cast in the form of a stress tensor whose deviatoric part is given by this expression. Okay, so essentially it's proportional to the tensor product of n times itself. And here there is a number, which we call S, the nematic order parameter is a number between 0 and 1, and tells how well ordered are these rods at the scale of a single volume element. So it's a local quantity. This object has a name. This name is Q tensor, and is a common object in the physics of liquid crystals. Okay, also tensor order parameter or nematic tensor. This number here, instead, alpha, is a constant which is proportional to the force exerted by these rods, so also to the velocity of the rods and to many other things. And is the active stress, is an important quantity in this field. Sometimes we just call it the activity, okay, for short, and somehow embodies all the biochemistry of this process. We often treat it as a constant. It's not necessarily constant, but nevertheless is at least uniform. in the system. Okay, so this will be one of the main objects of this talk. Now, the hydrodynamic equations of an active nematics can be derived very much based on phenomenological principles and some common sense. Okay, for instance, the momentum density of an active nematic liquid crystal is going to obey to the classic momentum density conservation law. Okay, so Navier-Stokes, with the difference that now, in addition to the viscous stress, okay, which represents viscous dissipation, and in addition to an elastic stress, because this liquid crystal can actually store energy into the form of distortion and release it into the form of a flow, there is also this active stress, which is the quantity that John pointed out in the previous slide, which is the contribution to the stress due to the fact that the molecules are, expanding and exerting forces on their surrounding. Okay, so it's actually very simple. At least it's a simple-minded way of modeling it. Now, Q is, of course, a field, is a dynamical variable, so it also needs an equation, a hydrodynamic equation. This, in 2 dimensions, can be constructed also phenomenologically in this way. Maybe it's a bit, less beautiful than the first one. Nevertheless, it embodies simple physics. In fact, it embodies 2 different processes. One is so-called flow alignment, is the fact that if you have a liquid crystal and you shear it, the nematic director will actually reorient compared to the flow direction. This can be captured by these 3 terms. And then there is also another term that describes the relaxational dynamics of this nematic phase. Namely, there is a free energy, and the system, in the absence of any forcing, internal or external, will relax in such a way to minimize such a free energy. This free energy is named after Landau and Dejean, and is the usual Mexican hat potential, just written in terms of the equation of the tensor Q. Okay. The reason why I'm somehow flashing these equations is, William, please. Yeah. Rotates, but it doesn't do like continuous orbit. It rotates, and if you do a shear experiment, it stabilizes at a precise angle. This is called the Leslie angle. The reason I'm flashing these equations is because I don't want you to remember them, but I want to point out that there are actually two length scales, okay, embodied in these equations. One is this one. It appears inside the free energy, and it's some kind of nematic persistence length, namely, it's a length scale that tells you how quickly the order parameter drops, for instance, in the neighborhood of a topological defects or a boundary layer and so on. But then there is another length scale, which is more important, that comes from the balance between the active torques, okay, exerted by this term that try to distort the system, and the elastic torques, okay, which are coming from the free energy, which instead tries to restore a uniformly aligned configuration. So there are these two effects that somehow compromise each other at a distance, which is given by this quantity LOA, and is proportional to the square root of K, which is the orientational stiffness of this nematic phase. How much do you have to, how much torque do you have to exert in order to rotate the director field divided by alpha? Okay. Now, the beauty of working with experimentalists in a lab is the fact that you can actually check these intuitions right away. And for instance, in the experiments by Zvon-Mirdojic, you can actually see both length scales simultaneously. This is epsilon, this is L sub A, okay, the active length scale. So one is the size of the defects, the other one is somehow the radius of curvature of these folds, of this deformation. The active length scale, as I said, is very important. And depending on how this length scale compares with the system size or the confinement length scale, you can actually explore all the zoology of active nematics. For instance, when the length scale is much smaller than the system size, typically the system is found in the lowest free energy state. Okay, so the configuration that minimizes the free energy. This might or may not be flowing, depending on the geometry, depending on the boundary condition, but is the ground state. Now, if you imagine to increase now the activity, okay, by putting, for instance, more of this biochemical fuel, eventually these active length scales will become of the order of the system size. The ground state becomes unstable, Okay, it becomes unstable to a state which is characterized by spontaneous distortion and typically by a flow, a laminar flow. Now, if you keep cranking up the activity, also this laminar flow will become unstable to something else. And here, the situation becomes a bit system-specific, okay? It depends on the parameter, depends on the model. But for instance, it's possible to observe a periodic flow, so a transition from laminar to periodic, and eventually this periodic flow somehow degenerates into a turbulent flow, a chaotic flow, okay? I made these lines sharp because these are sharp transition. You can also calculate the transition point. This one instead seems to be something much more smooth, more like a crossover from periodic to turbulent. So the story that I will tell you is essentially in this regime, and especially when the active length scale is much smaller than the system side, but much larger than the other length scale I showed, than the sides of the defects. Okay, so there is this somehow sweet spot, which nevertheless is very interesting. Now here is somehow some simulation to show how these flows look like. And I must say I'm very embarrassed to show the simulation after what I saw yesterday, you know, galaxy and so on. This looks very simple, but that's all I have. But here is a plot of the vorticity of the velocity field, the arrows superimposed to a heat map of the vorticity field. So, you know, red and blue are positive and negative vorticity. This instead is a common way in the physics of liquid crystal to visualize the orientation of the director. It's called a Schlieren texture, and is essentially what you would see if you put your nematic under a cross polarizer. Okay, these little dots that you might notice, blue and red, are topological defects. So are points in this configuration where the director orientation is undefined. Okay, and there are a lot of them in these chaotic turbulent flows. All right, so the first thing that one can do, I mean, first to check whether the name turbulence, active turbulence, defect turbulence is an appropriate one, is to calculate the probability distribution function of the velocity and the vorticity. And sorry, I forgot to put the labels here, but these are the x and y component of the velocity field. Everything is strictly two-dimensional here, and this is the vorticity field, okay? And as you can see, the velocity field is very much a Gaussian, okay? Whereas on the other end, the vorticity field has these prominent non-Gaussian tails. And you know, this is one of the reasons why I think turbulence is not a bad name for this type of systems, because I believe that this is exactly what you see in two-dimensional inertial turbulence. However, if we want to make progress, okay, we need to understand a little bit about the geometry, okay, of this chaotic flow. And one way to do that, maybe not the only one, is to introduce what I like to call a sure. I don't have it. I mean, I think I have it somewhere, but not on these slides. Yeah. To be honest, now I don't remember. Yeah. Also because I mean I'm more familiar with the statistics of the velocity gradients in three-dimensional turbulence, and in 2D turbulence, I actually am not sure. Should be regular. So yeah, certainly there is no intermittency, and so you would see, so all the moments would scale in a predictable way. All right, so this vortex areal density is a function, let's call it n of a, such that the product of n of a times dA is the number of vortices whose area is in between A and A plus dA. Okay, so it's a simple vortex areal density. This quantity can be measured, and this is the result. Okay, so this is a paper that I wrote in 2015. So as you can see, so this is, by the way, is a log linear scale. Okay, so a straight line indicates an exponential distribution. So there is indeed a range of scales where this density is exponentially distributed as the following functional form. Okay, now in this expression, capital N is the total number of vortices, which is not a conserved quantity, and Z instead is just an integration constant. This is just a bit of terminology. But more importantly, this exponential distribution identifies two area scales, okay, two peculiar areas, which are indeed the minimal area here when the distribution stops being exponential, and the typical slope of this exponential line, which I call A star. Okay. Now, remember what I told you earlier. Okay, so we have these two different areas, and we want to link them with the parameters of the model. Because there is a single important length scale in the problem in this range, I mean, far away from the defect core, well, then it's most reasonable to assume that both these areas are roughly similar and proportional to the squared active length scale, so the ratio between these two constants. Okay, so this is just dimensional analysis, really. How about vorticity? Okay, these vortices also have a vorticity. Now, if you look at the equation, especially if you cast the equations in terms of vorticity rather than velocity, then you immediately realize that the rate at which this active stress is injecting vorticity into the flow is given by the ratio between these two constants, alpha divided by eta, eta being the shear viscosity. Okay. Now, interestingly enough, this quantity is independent on the vortex area. Okay, at least if you do this back-of-the-envelope calculation. So the simple picture which is kind of emerging from this numerical data is that perhaps we can think of a turbulent flow in active pneumatics as some kind of collection of vortices, okay, spanning a number of scales, but whose average area is proportional to the squared active length scale, and whose vorticity is proportional to the ratio between the activity divided by the shear viscosity. Okay, so this is somehow the picture that one can try to infer by looking at this data.
In two-dimensional turbulence, ordinary one, you would get vortices when there is fluid that is devoid of vorticity, and then you have the next vortex, so you can make a vortex census and all this. But is that also the case in your application?
That's a good question. No, it's not. It's not the same actually. Okay, that's a good question, but it's a bit of a technical one. So there are criteria for that. One that is used, for instance, in meteorology, uses a field called the Kubo-Weiss field, which is roughly related to the Lyapunov exponent of a tracer particle, which is advected by this flow. So based on that quantity, you can determine whether you are on a vortex or you are in this shear zone. Okay, and by mapping this quantity and do some image analysis, it's possible to reconstruct the shape and the area of these vortices.
Is that how those distributions work out?
Yeah. Exactly. So it's possible to now verify to test this picture by, again, looking at the numerical data, and it kind of works. So these are the two areas that I showed as a function of the square activeness scale. They collapse on top of one another. They are proportional to L sub A square. This is the vorticity of a vortex as a function of the area. I told you that it should be independent of the area. I don't know, maybe if you look carefully, you will see that there is a little bit of slope here, okay? But I would argue that this is weakly dependent, okay? All right, so we are in good shape. We can use somehow this, numerical evidence and this bit of intuition in order to construct some kind of ensemble of vortices, okay? So rather than, if you want to make progress, rather than looking at the flow in the traditional way, what we can do is to imagine that such a flow consists of an ensemble of vortices whose area is exponentially distributed, whose average area is proportional to k over alpha, and whose vorticity is independent on the area, roughly proportional to alpha over the shear viscosity. Okay. Now, this is not just somehow a way to make sense of things, okay, like a conceptual framework. This is also a calculation tool, because now what we can do is to replace the special averages that you would like to calculate when you deal with turbulence with the ensemble averages. Okay, so far this is a work hypothesis. It's not tested. So let's see how it works when we apply this strategy. A few things that you can calculate, and it will make sense to do it, is to how the entropy, okay, so the square vorticity, and the energy, the kinetic energy, scale with the activity in the system. Okay, it makes sense to calculate these quantities. And if you do it, you realize that the entropy scales quadratically, okay, with the activity, while the energy scales linearly with the activity. So this is what I would expect. This is a bit less trivial, okay? And the reason is a bit less trivial is because if the kinetic energy scales linearly with the activity, it means that the velocity, okay, scales like a square root of the activity, okay? And this is not what I would have expected, naively, because somehow the forcing term that drives the flow is proportional to alpha, and so you would expect that also the velocity would be proportional to alpha. And indeed, that's exactly what happens in active laminar flow, okay, but not in turbulent flow. Let's see if we can make sense of that using this ensemble of vortices. So replacing these special averages with ensemble averages, one can calculate the kinetic energy is equal to this expression. Now, this is independent on the area, so this is a very simple integral, okay, and is equal to the omega square times the ratio between the second and first moment of this distribution, which is proportional to alpha. And analogously, for the entropy, you get something similar, which is proportional to the square of alpha, consistently with the observation. This is a bit dry, I understand, but it's actually a very simple way to, intuitive way to understand these properties. These vortices, okay, have a size that scales like 1 over the square root of alpha. And the angular velocity, let's put it that way, that scales proportional to alpha. And if you just apply, rigid body rotation type of approach, you will convince yourself that the velocity should be proportional to the product of these two quantities, which indeed scales with the square root of alpha. So, the bottom line is that if you increase the activity, the vortices become faster but smaller, okay? And so this affects the kinetic energy. All right, that was a simple one, I have to admit. Now, let's try to do something a bit more complicated. Let's look at the spectral densities, okay? So this is the holy grail of any, theoretical work on turbulence. As a reminder, so the energy spectrum is defined from this expression. The entropy spectrum is defined from this expression. It is actually trivially related to the energy spectrum. Just for comparison, this is what happens in two-dimensional turbulence. So in two-dimensional turbulence, you have altogether 4 exponents, which are universal. And you have 2 of them because there is a direct entropy cascade and an inverse energy cascade. Okay, and each of them has a signature on the spectra. So for instance, for the energy, you have Kolmogorov type of scaling to small k value, k being the wave number, and k to the -3 to large k value. This is the energy. For the entropy, on the other end, you have k to the 1/3, okay, for small k values, and k to the -1 for large k values. Okay, so this is what happens for inertial turbulence. Now, let's see active turbulence. So here there is a difficulty, okay? And the difficulty is that you have to somehow parameterize this vorticity field. And one way to do that, again, maybe not the only one, maybe not the best, is to assume that this vorticity comes from a superposition of vorticity of a single vortex, okay? And this can be written as this constant, omega b, the vorticity of each single vortex that we know does not depend on the area and has a simple structure, times, this is a structure function that tells essential information about the geometry of a vortex, okay? How the vorticity changes if you go far away from the center of the vortex. Now, we don't know this function, okay? And by this numerical simulation, we can also argue that the geometry of these vortices is kind of complicated. Nevertheless, I'm a physicist, and therefore I like spherical chaos and also, circular vortices, okay? So if you assume that your vortex is just a disk that is kind of rotating, okay, then it means that this structure function is a step function, okay, which is equal to 1 inside the vortex and 0 outside the vortex. This is a simple parameterization of this vorticity field. Next, what you can do, you can use this parameterization to calculate the ensemble average under the assumption that the vortices are spatially uncorrelated. So this is a mean field assumption, and it's the best that I was able to do at that time. The spectrum for the entropy that you calculate using this approach is kind of convoluted, okay? It contains a couple of Bessel functions, a Gaussian, so it's not really simple to decipher. Nevertheless, has very simple and clear asymptotic behavior. It scales like k for small k values. So this, Greek kappa is just a dimensional version of the wave number, and it scales like k to the minus 2 for large k values. And as a consequence of that, the energy spectrum scales like k to the minus 1 for small k values and k to the minus 4 for large k values. So here are the 4 exponents, okay? How do they compare with the numerical simulations? And again, I need to somehow confess my embarrassment, because in 2015 that was the best that I was able to do. 256 by 256 square grid, very small. So I didn't even try to actually feed the small wave number region. And this is just, a line that shows the analytical prediction. You can argue that this is, kind of consistent, or at least not obviously wrong, right? However, thanks, fortunately for me, other people devised a better way to do this simulation. So this is a paper by Alert, Joanny, and Casademun that was, you can find it on the archive. And in this case, they managed to go on much larger sizes, also using a clever parameterization of this nematic field. And here, the 4 exponents actually, are manifest. Okay, so they are exactly what predicted by the theory. Let me add a little bit of information about the defects. We heard about the defects that play an important role in this active turbulence. I just want to explain why they play an important role. Because somehow, as a consequence of these active stresses, okay, that are injecting, force inside the system, the defects are able to actually generate flows. And these flows are very closely related to the geometry of the director field around the defect. And this geometry is actually very robust, okay, because these defects are topological features, singularities in the director field. This is why we call it topological, by the way. So for instance, a +1 defect, so this is called a +1 disclination, this type of texture, and will produce a couple of counter-rotating vortices. This is a -1 disclination and will produce more vortices. The bottom line is that the statistics of the vortices and that of the defects is actually the same. Okay, so if you know one, you can infer the other. Although I'm not sure that from the defects you actually can infer the vortices. Even though I see recent work by the group of Dunkel and Nita Fagri that seems to actually demonstrate also that you can go in the other direction. All right, so if you know that, you can actually pretty much figure out all the properties of the defects. For instance, you can see that the number of defects actually scales linearly with the activity. When I was working on this problem, this was somehow one of the open questions, how did it scale? Now, so this was somehow this theory of 2015. The first experimental group that took the trouble to actually test it was the group of Francesc Agués in Barcelona. They did the same analysis on these microtubule Keynesian systems, gently provided by the Dogic lab, and they did find the exponential distribution of the area. But nevertheless, they only use one value of the activity, okay? So we don't know how this line moves up and down, if you change the activity, and they didn't measure the vorticity, okay? That's very important. If you know this distribution and you know that the vorticity does not depend on the area, then essentially you can calculate everything about the flow. So more recently, Zvonimir, together with the student Linnea Lemma and Stephen Degamp, did a more sophisticated version of these experiments. They tried a bunch of different activity values and managed to confirm more. Now, if you ignore these 2 lines here, I will explain you why you have to ignore them, you can see that indeed the exponential distribution is recovered, and the fact that the vorticity does not depend on the area is actually very, very beautifully recovered, I would say. Nevertheless, Zvonimir and Linnea also noticed that if you go to very small activity values, well, then the exponential distribution is not that exponential anymore, okay? And why is that? Well, there can be many different reasons. Too close to onset, for instance, or not enough statistics, or perhaps for small activity values there are other effects that come into play, okay, and break down the universality. Last but not least, this is a recent experiment of the group of Pascal Silberzan at Curie, and to my immense surprise, they managed to recover the very much same features even in epithelial cells. Okay, that's again the probability distribution function of the vortex area, and those are the vorticity for counter-rotating clockwise and counterclockwise vortices. Okay, so this seems to be really a universal feature in the traditional sense of active turbulence. I could speculate about the origin of this experiential distribution, but it's actually so simple that I won't do it. Instead, what I would like to do is to thanks, in addition to finding agency, my experimental collaborators who are really, have done a tremendous amount of work, okay, and I'm really grateful for that, and of course you for your attention.
We have time for a couple of questions, please.
Have you thought about more how to include correlations? Of course, it's impressive the agreement you get even without that, but I would at least think two vortices can't be on top of each other, so there's going to be an anti-correlation, at small scales. Maybe that's already in there, but...
Yeah, so let's see if I have the slide here. No, I actually removed it. So the correlation between the vortices it becomes evident if you calculate the vorticity correlation function, okay? Because you can see very clearly that it agrees with this mean field theory up to the way when you have you move far one typical side of the vortex, and then it becomes anti-correlated because the positive vortex is surrounded by negative vortices just for continuity. So yes, I mean, there is definitely a correlation. Doesn't seem to affect not just the small scale behavior, but even the large scale behavior. And this is kind of surprising, okay? So I don't know why, but it's certainly something that it would be interesting to look at.
One more, please.
Is there in these systems any notion of permanence of vorticity, like being relatively constant following the fluid? Or is it flickers like a light bulb, goes on off?
I mean, I would say that it does flicker like light bulbs. I mean, so what you have is that the average number of vortices stays the same. Okay, so if you count the vortices and you average over the sample, you'll see that indeed this number is constant. And this is one of the things that you can actually use to make sense of this exponential distribution by using Boltzmann counting at the end of the day. But they are very non-persistent. I mean, they appear and disappear continuously, just like the defects. Yeah.
Okay, I think we postpone other questions to the discussion. Let's thank Luca again.
Thank you.
Hi, everyone. My name is Junhui Liu. I'm a graduate student working with Nikta Fakhri in MIT. So today I'm very excited about telling you the story about a fascinating signaling event that takes place inside the living cell, and where the protein of a nanometer size could organize and orchestrate into waves that propagate into hundreds of microns in scale. So what's even more fascinating about this whole process is that we can use turbulence to understand it. I want to start by first thanking everyone that is involved in this project, especially Pearson and Zehan that have also come to this conference. Pearson from Professor Jorn Dunkel's group is also going to shortly talk the second part of the story. So the signal event I just showed you takes place inside the starfish oocyte. Starfish oocytes are large, round, and transparent cells having a typical diameter of around 150 micron, which is huge compared to the size of the signaling protein, Rho protein, that are known to guide important cellular function during the early oocyte development. So the Rho protein in the oocyte exists in two conformations, an inactive conformation in the cytosol and an active conformation on the membrane. Transition between the two conformations are catalyzed by enzymes in the oocyte. So despite the huge size difference between the Rho protein and the starfish oocyte, during early development, the concentration of the Rho protein in its active conformation often needs to be coordinated on the cell level. An example is the initiation and propagation of spiral waves in the active Rho protein from one side to the other of the side of the cell when we put extra activation enzyme GAF into the oocyte. Not only could the spirals form and propagate on the cell level size, it could also later organize into those complex wave propagation patterns with a sustainable length scale on the whole cell level, but also maintain really rapid local concentration turnover. So the video here are the fluorescence intensity of the active rho reporter that we visualize using live cell imaging. So specifically, we also know that active rho does not just migrate on the membrane. Formation of such cell-level signaling events is a combination of both the reaction events involving the rho exchange between the cytosol reservoir and the membrane reservoir, and also the diffusion events where rho both diffuse on the membrane and in the cytosol. The GAF molecule activates a self-catalyzed transition between the inactive conformation to the active conformation on the membrane. We know that such reaction diffusion kinetics embeds a limit cycle in the rho species phase space, which corresponds to the local oscillatory dynamics that we already observe in the active rho species. What's even more intriguing is that experimentally, we could indeed tune the GAF level in different cells, and what we get is very different complex wave propagation patterns. Those videos are generated independently from different oocytes. We term these patterns as different independent steady states because they represent our access to a variety of non-equilibrium steady state with continual injection of energy to sustain those signaling patterns in living cells. Each of those states maintains their typical wavelengths ranging from 15 to 30 micron, and also their typical temporal oscillation period ranging from 50 to 120 seconds. So despite that those non-equilibrium steady states exhibit very different spatial wavelengths as well as temporal oscillation period, the fact that they all locally oscillate permits us to use phase as the other parameter. So experimentally, imaging data are taken on a pixel grid, with each pixel being half a micron in length and width. When we record the fluorescence intensity time series, we realize that such local oscillatory dynamics persist even in a single pixel resolution. We can then use a time delay embedding algorithm to extract a phase value at every time point. And the extracted phase has a range of -pi to pi. In that way, we can extract a phase time series corresponding to every intensity time series we have at every single pixel. And when we combine them together, we get our reconstructed phase field. When we put the protein concentration field and the reconstructed phase field side by side together, we can see that there exists a dense collection of topological defects in the phase field, corresponding to the spiral course in the wave propagation pattern. Those defects, as discontinuity point in the phase field, turns out to belong to two classes, 1D number +1 and 1D number -1, corresponding to the clockwise rotating and the counterclockwise rotating centers in the phase field. In our field of view, those two types of defects always maintain similar density. When we track the defect trajectory as point object moving from frame to frame, we can also see that they have intriguing dynamics. At any instantaneous time, there always exist the newly created pairs of +1 and -1, and also the newly annihilated pairs. Defects that are long-lived are not positioned close enough to any of its oppositely charged partner, and they meanders around with a linearly growing mean square displacement. When we calculate the total defect density as a function of time, we realize that across all those steady states, they all maintain distinct defect density, but they are all steady despite such continual flow of creation and annihilation events happening. So when we generate more independent steady states and we quantify those defect statistics, we realize that such defect density very nicely scale with the wave number we could extract from the protein propagation wave pattern. So these robust defect statistics then suggest to us that there might exist a specific mechanism across these states that guides the defect organization. So to resolve such a potential organization mechanism, we turn to a view in the phase velocity field as a way to quantify the phase coupling between the neighboring oscillators on the membrane. So the phase velocity field, here defined as the gradient of the phase field, characterizes a rich existence of the vortices, highly localized around center as the phase defects. The feature that those vortices has highly localized structure and also have very similar appearance reminds us of certain vortex structures in other two-dimensional turbulence systems. For example, probably a quantum turbulence state. In fact, we can retrieve a physical interpretation of our phase velocity field by making this analogy to superfluid. So in superfluid system, there is this connection between the fluid velocity and the phase velocity established by a constant. If we change that h bar over m constant by the diffusion constant of active rho protein on the membrane, then what we can simply retrieve is the fluid velocity in our protein wave system, which now corresponds to the information propagation velocity encoded in the protein concentration waves. So from that perspective, we could aim to analyze the organization of the phase velocity field with a variety of length scale analysis, and we can aim to characterize those protein wave propagation patterns as emergent state arising from the protein component interaction. So we can first start with a global characterization of the phase velocity field. Visualized here is the streamline representation of the phase velocity field and also the colored vorticity field of the phase velocity. We can see that consistent with our initial observation, the high vorticity magnitude are very tightly concentrated around those vortex centers that are similar for all the vortices on the membrane. We can first quantify as a collective statistics about the tendency of vortices to exchange partners. So at any instantaneous time, we could identify the vortex and anti-vortex pair that are closestly positioned to each other compared to all the other surrounding vortices, and determine whether such pairing are persistent over a consecutive number of frames or transient, that they change partners quickly. It turns out that the fraction of persistent dipole pairs remain uniformly rather low across all the steady states we have, which suggests to us that the vortices in the phase velocity field are characterized uniformly by a low fraction of the bound vortex-anti-vortex pairs, which is reminiscent of a disordered phase in the BKT phases. We can also measure the energy of those disordered states by defining an effective kinetic energy by taking the integral of the squared phase velocity on the two-dimensional membrane and compare for every single steady state with a measure of effective entropy defined as the integral of squared vorticity on the membrane. What we realize is that across different steady states, there exists a really nice linear relationship between the two quantities. The linear relationship between the kinetic energy and the entropy has previously been described by Jorn in the observation and theoretical description of two-dimensional active bacteria turbulence. Then we realize that our data on the global scale has some turbulent character. When we zoom in to an intermediate length scale, we can see that the pairwise collision between vortex and anti-vortex happens from time to time and significantly affected the individual vortex motion. We can then try to characterize such collision statistics by locating all the pairs of vortex and anti-vortex that eventually annihilated and plot their relative speed as a function of their pairwise distance. When we take in all the statistics we have of more than 6,000 vortex pairs from all different steady states, we realize that there still existed this substantial acceleration as the pairwise distance decreased. We can also decompose the relative speed into tangential and radial component, and this is when we realize that such acceleration is definitively contributed to by the radial components, which is indicative of a conserved vortex-anti-vortex attraction across the states. We can also notice there exists a transition in the noise level embedded in those curves as the pairwise distance decreases. This could also be explained in the pairwise interaction picture, since this could be interpreted as a many-body to two-body interaction transition. When the pair vortices are far apart from each other, they interact with multiple partners, thus giving this noisy dynamics in this range. But when they move closer with each other, they experience a more conserved pairwise interaction thus giving this conserved acceleration across states. In fact, when we quantify the length scale on which such transition happens, around 6-7 micron, this is also consistent with the length scale we could extract from the previous kinetic energy and entropy linear relation. So we can then go into a smaller length scale predicted by the two-body interaction range about the pairwise pre-annihilation dynamics. This corresponds to the spiral core annihilation in the protein concentration field. Visualized here is the streamline representation of the phase velocity field, where we can already see there exists a strong compression of those phase velocity streamlines when the pair vortices approach each other. We can quantify such pre-annihilation statistics by plotting their pairwise distance and also the radial relative velocity as a function of time before annihilation. And what we realize is that across different steady states, there indeed existed this conserved acceleration dynamics. It turns out that such conserved acceleration dynamics gives rise to a also conserved velocity statistics. So when we quantify the distribution of the normalized vortex speed, we realize that different steady states exhibit not only a similar parallel distributed tail at the high speed end, exponent of those parallel distribution also exhibits very common value around -3, which to us is reminiscent of the feature that Professor Nigel-Goldenfield has suggested in the collection of two-dimensional pairwise interacting topological defects. And all these velocity statistics suggest to us that our vortices exhibit a generic two-dimensional vortex-vortex interaction. Apart from those multi-length scale analysis, we could also mathematically quantify the turbulence. So by looking at the braiding motion of the space-time trajectory of the defects, we could quantify the braiding factor, that is how entangled those space-time trajectories are, and also the braiding exponent, that is how fast such entanglement grows as a function of time. What we realize is that for a single steady state, by looking at those braiding exponent, we indeed could retrieve the positive exponent over a range of trajectory number, which to us suggested that the defects are indeed moving chaotically. What's even more intriguing is that when we calculate such braiding exponent curve for different steady states, they have similar shape but different magnitude, which by the braiding statistics definition represents a low to high order of increasing rates of local information loss. And when we map to the protein signaling pattern, indeed, it represents a low to intermediate and then high level of local information scrambling. So the turbulence statistics could also be reflected by the vortex size. So for any single steady state, we could conveniently quantify the vortex size by constructing the Voronoi tessellation at any instantaneous time and plot their tessellation area statistics. As we mentioned before, those different steady states have very different defect density, up to 10-fold difference. So this naturally leads to a wild distribution range on the tessellation area axis. But when we try to normalize those distributions using the mean area per vortex occupied, we realize again, surprisingly, that there is this collapsing distribution that all the different steady states share, and which also features a prominent exponential decay, which again we find to be surprisingly matching with what Professor Luca Giammi just mentioned. So as a conclusion to the first part of the story, experimentally we have generated accessible non-equilibrium steady states of those signaling protein waves. By looking in their phase velocity field, we realize that they exhibit features of statistics reminiscent of two-dimensional turbulence, including linear relation between energy and entropy, minus rescaling of velocity distribution, and also exponential decay in the vortex size distribution. We realize, as a conclusion, that we can use generic two-dimensional vortex-vortex interaction underlying those protein patterns as an interpretation. And we would also like to suggest that this is a novel approach to explore the similarity and difference between defect dynamics and information propagation. And we want to see that extended to more living and non-living systems. I think this is the end of the story. Thank you for your attention, and I'll take any questions.
Clarify how do you compute this grading factor?
Oh, sure. So it's basically by, okay, let me switch to here. So it's by quantifying at every time point the crossing between the long-lived defect. So you have like the trajectory group. Yes, the trajectory cross.
It would turn around like in a superfluid. So this
Yes. Yes, so there is this measuring topological chaos paper.
number, I think there's a name like this.
The winding number.
Yeah, when a particle goes around another particle. Related?
Yeah, I guess we use the word winding number mostly for the topological defect charge. So I don't really want to get that confused. Here we're looking at the center of those vortices, those topological defects, and looking at the trajectory, how they cross over. Yeah.
Yes, please.
I just wanted to point out that your two-dimensional vortex statistics is the two-dimensional analog of what I described in my talk as three-dimensional vortex statistics. And in my case, there were circulations around the handle of the vortex structure. In your case, it's circulation around the point vortex, but in both cases, there are winding numbers which are quantized.
Yes,
So
yes.
it was very interesting to actually compare my theory in two-dimensional version with your experiment.
Yeah, that'll be super interesting. Thank you for pointing that out.
Have you taken a census of your critical points in your flow field?
The critical points?
Yeah, like hyperbolic and elliptical and saddle points.
So that I think I'm not totally sure about what that refers to. Like in the it's not a hydrodynamic system, so about the criticality in the flow. We have something around criticality in terms of vortex organization, but we can talk more. I don't think that's probably what you mean.
No, it's like points in the flow where the velocity is 0. Right?
Oh, yeah, I see.
Do you know if it follows the Euler index theorem? Have you checked that?
We haven't looked at that, but I'm happy to talk more in case if I misunderstood.
More questions? If not, then let's thank the speaker again.
So I'll be talking about the more theoretical side of the previous study. In particular, this is a conference, we're arguably looking at universality. So I'm going to be talking about, some attempts we've made to identify universal dynamics in these turbulent vortex statistics that were just discussed. So just a brief background, since I know there are a lot of people who are not biologists at all in the room. People have been mathematically studying reaction diffusion systems in biology for about half a century now. Traditionally we go back to the great Turing, who in a seminal paper called The Chemical Basis of Morphogenesis really started looking at equations of this form and how they're really essential for any kind of spatio-temporal organization in life. Now for the last 30 or so years, people have been moving from studying these, classic pattern formation examples of Turing to what's called topological or defect-mediated turbulence, which is reaction diffusion systems that exist in a highly chaotic regime. Now, on Monday, Nigel shared some really good wisdom, I think, from Feynman about, extracting qualitative information from equations and how this is sort of the future, the next step for us theorists. But that's sort of conditional on having equations to begin with. And one of the great challenges of this field from a theory perspective is our vast ignorance, really, of the precise reaction kinetics going on. So this is this cartoon we were using in the previous talk and elsewhere, depicting our system. You have these, central protein, what we're imaging, this rho GTP, which can be phosphorylated or dephosphorylated to rho GDP, and it interacts with two enzymes, GEF and GAP, one of which activates it, the other deactivates it. But really, like all things in life, there are many other proteins simultaneously flowing through the membrane and the cytosol. And it's really not clear, one, how many proteins are actually involved in any given reaction network, and two, which proteins are interacting with which other proteins. So to highlight this, here are 4 different sketches from 4 papers published in the last couple of years, each of which was studying the same reaction diffusion system as we are. And you can see that each of them kind of identifies a different subset of key reaction diffusion interactions, various positive and negative feedback loops between different parts of the system. And each one of these different cartoons would represent a different set of nonlinearities in a continuum model of the system. So as a consequence, it becomes very difficult to instead look at a single model, and thus we seek instead looking for something a little bit more universal. Now, to do that, we ask, what do we actually know about this system? Well, one thing we're pretty confident on is that this system undergoes a Hopf bifurcation when we're perturbing it from, this is our starfish oocyte and its wild type, where there's just a single traveling wave, it's excitable. And as some parameter is changed to activate these spiral waves, it goes to this oscillatory mode instead. And it's believed this is Hopf bifurcation. And as many of us know, there is a good universal model for the behavior of an isotropic system near Hopf bifurcation. And that's the complex Ginzburg-Landau equation. And so for a given reaction diffusion network, you can map this to a complex Ginzburg-Landau. You do this, say you take this one, just imagine we have our rho GTP and there's some positive feedback loop with rho GEF, this enzyme. And you can imagine this might be represented by a reaction diffusion system of this form, a fusion term, some sort of cooperative activation of the rho GDP to rho GTP, and then this decays via rho GAP, which isn't mentioned here. And then one can find the fixed point, one can identify the Hopf bifurcation, and proceed through a very cookbook analysis to derive from this equation, for a given set of reaction rates, a corresponding, value for these 2 parameters in the complex Ginzburg-Landau model. So in this case, if we set k gap equal to 10, do this procedure, you get something that maps to Ginzburg-Landau with b equals minus 1.9, c equals 1.5. In this case, it's actually a pretty nice match. For our model I chose pretty much arbitrarily. This is the simulations for those 2 parameters I mentioned, and this is one of our experimental oocytes. These are the phase maps. So it's okay, eyeballing this doesn't look that bad. And for other experimental data points, there are places in the Ginzburg-Landau phase space that visually look pretty similar. So that's okay, good sanity check, good first sign. But more importantly, so as I say, we're not trying to demonstrate the validity of any particular reaction diffusion model. We're asking, does complex Ginzburg-Landau, represent a universality class into which all of our defect statistics previously discussed fall? And the answer is, it's not a bad match. So here's that experimental data from before. This is the velocity scaling of our point defects. It's vaguely -3, though it's noisy, that exponent scaling in the asymptotic limit. And you can do the same thing for, run a bunch of simulations of Ginzburg-Landau at different points in the parameter space. So these are all different morphologies, as I showed here. But in all cases, you can, following the same procedure, identify point defects at the spiral cores and calculate their velocities, and you get very similar scaling. The exponent varies a little bit depending on the value of the exact point in the parameter space, but in all cases, it's power loss asymptotic behavior, which is interesting. And then further, if we go to this looking at the vortex area distribution, which we've heard a lot about today in various talks, ditto. So again, the experimental data from before, and this is what you see at various points in the Ginzburg-Landau space. And you again get this exponential tail, semi-log plots or exponential tail. I should mention that as before, and in differently from Lucas' talk, we've collapsed this data by dividing everything by the mean area. So that's why the exact scaling is the same for all curves in both cases. But in all other respects, this is very consistent with what we were seeing also in the active dramatics. And so the takeaway from this is it seems like the defect scaling behavior we're seeing in our system holds true for a really wide region of the complex Ginzburg-Landau parameter space. And that suggests that it would also hold true for a wide number of possible alternative reaction diffusion networks, which might be our, including the one that hypothetically would work for our system. So that's very interesting to us. But the other question we're asking is, we're interested in these defects. We're interested in kind of the self-sufficiency of these defects as a way of explaining the dynamics of the system. So what I'd like to compare this data to is, instead of another continuum model, another universal continuum model, let's ask, are these statistics also emergent when you have just a simple system of interacting point particles? So here we have, probably the simplest interacting vortex model you can come up with, which is this one, which I believe goes back to Onsager, where you just have defects on the surface of a sphere, in this case, interacting with logarithmic potential, according to this Hamiltonian. Defects of plus or minus 1 charge, as we have in our system. And, this represents, again, probably the simplest case you can have for one of these systems, point particles interacting. There we go. And once again, you observe here very similar statistics. So again, experiments, this is the CGL results from the previous slide, and this is the results you get out of this point vortex model of interacting points on a sphere. And in all cases, you have this exponential tail for the area distribution. The crucial difference is there's difference in slope. Now, this is in all cases, they scale, the characteristic area is normalized out. So instead, there's some additional factor that's different in these two cases. These two are pretty close, but between the two theory models and the data. But nonetheless, the functional form is the same in all three cases. And likewise, when you look at the form of the velocity statistics, so in this case, again, as before, we look at, on the surface of a sphere, the statistics of the velocity field, or velocity of each defect, and calculate the PDF, you get power law scaling. Now, in contrast to the complex Ginzburg-Landau case, you actually can get a variety of different scaling exponents, and this seems to be strictly dependent on the energy of the Hamiltonian. So this is a Hamiltonian system, the point vortex model. You specify the energy when you initiate simulations, and you let it run. And so what we actually see is, if you plot the density of states, the value that's closest to the -3 scaling you get here corresponds to more or less this light blue curve, which is somewhat less than the mean most probable energy. Now, I think a lot of you are probably familiar with the point vortex model, but just as a quick refresher, it's got an interesting feature where there's this transition between, in the low energy regime, pairwise interactions and transitions. Pairwise interactions, transitions to effectively independent particles. And then the high energy regime goes into these phase separations where all the like particles of one sign clump and all the ones of the opposite sign clump together in a separate cluster. So what this is telling us is the velocity statistics from this point vortex model that most closely match our experiments is somewhat in this intermediate regime, somewhere between here and here, which is to say most of the particles might be independent, but some of them are pairwise. And if you remember from Xinghui's talk, that's actually pretty consistent with what we see. It just happens that, perhaps 20% of the particles of the vortices we observe in experiment are in fact in pairwise bound states, and the rest are unbound. Likewise, again, in all three cases, the exponents match up reasonably, given that this is especially a particularly noisy exponent. So I feel like the takeaway from this, the important takeaway is from this point vortex model, is that the dynamics we observe in this reaction diffusion system it's sufficient to have just interactions between the topological defects. The topological defect interactions are sufficient to explain most of the key statistical behavior we were observing without the need for the broader continuum model. And that's exciting, because that's the real reason one ever is interested in topology in the first place, is that it's a way of reducing a more complicated system to something less complicated. So just to summarize what I had to talk about, the vortices within reaction diffusion systems in vivo, they exhibit robust asymptotic statistics that don't depend on the specific morphology of the chemical reaction spirals that we're observing. In a variety of states, they have the same statistics. And these statistics are matched by a complex Ginzburg-Landau equation, which one would reasonably expect to be a universal model to cover the sorts of reaction kinetics we expect to exist in the system. And finally, an interacting point vortex model is sufficient to recapitulate these statistics, suggesting that the system is in fact reducible to just a set of interacting defects rather than a more broad, more complicated continuum system. So with that, I'd just like to thank Jinghui, of course, Zerhan, who's also here, and our other collaborator, Melis, and then Professor Jorn Dunkel and Nick DeFakri from MIT, who are my co-advisors. And thanks for being here.
Thanks for being perfectly on time for this question. Yes, please.
Yeah, so you're pointing at me, is that right? Yeah. I don't completely understand your last point, because I thought the creation and annihilation was a substantial part of the dynamics that we saw, and that won't occur in the point vortex.
That's true. That's true. So obviously the point vortex model doesn't have creation and annihilation, but it does have pairing of vortices, and that's what I'm referring to. It's the fact that again, the point vortex model is sufficient. How do I go back on this? It does preserve total charge.
Yes.
Obviously, yeah. But what it does do is it gives you the same statistics, and it also can coincidentally, perhaps, happens to exist in a regime with a roughly same number of charged bound pairs. Now,
I'm not criticizing. It did seem as though from the movies we saw from the previous talk that was a really quite substantial part of the dynamics. That's all.
And that's
it was just a
absolutely
comment.
true.
It's not really.
I wouldn't propose this as an actual model of the system. The key idea here is just that turbulent, defect interactions are sufficient to explain these dynamical features without the need for a broader reaction diffusion model.
I'm going to take it off on that.
Mm-hmm.
So you. If you turn on your microphone.
Right side.
So taking off on that. Excellent. There we go. So you have creation and annihilation, but maybe the more important part of that is that you have attraction and you probably have repulsion between, like sign things that you don't have in the point vortex model. So I'm wondering if you endowed the point vortex model with a couple of extra interactions that might pick out the V to the minus.
Well, so again, we chose the point vortex model because it's simple, but the obvious next step for this, I think, is there are some known solutions for vortex interactions for actual reaction diffusion system. So I think there's a known solution for if you have two vortices in the complex Ginzburg-Landau, they'll have a velocity interaction that's exponentially decaying. And it's an attractive one too.
Okay.
So that would
But
be the one to try.
This by itself doesn't seem that strong because you are able to go through a continuum of different scaling laws, right? And you find one which looks like v to the -3.
Oh, of course,
Right,
yeah.
so-
But again, we're looking for sufficiency in existence so far, not like for necessary.
Okay.
I have the same comment made with the previous talk, but I also have a question. This statistics, you have asymptotic statistics which emerges after stabilization or long-term simulation of the complex Landau-Ginzburg or other equations, but it's not exponential of minus beta times this point vortex Hamiltonian. It's not Gibbs distribution with this point vortex Hamiltonian. It's something more complex, right?
Yeah, presumably, I think.
Do you have any idea what it is?
For the actual system, you mean?
Well, you have statistics.
Yes.
Is described by probability distribution. In your case, degrees of freedom are rather simple, just vortices.
Yes.
So, what is effective PDF for the ensemble of vortices? If it's not ideal gas with this.
For the entire ensemble, as opposed to just like the velocity statistics or something like that, we do not know the exact full ensemble.
That theory is done. What you see there is that Onsager, this is Kirchhoff's point vortex model. What Onsager did point out that there's positive and negative temperature states, which correspond to the left and right
Right.
of the picture. If you ever couple the system as an open system to something with a positive temperature, you will always be in the regime that you sort of observe, because the negative temperature state you wouldn't be able to realize that. So the branch that you find best agreement is probably because the system is actually open, right? It can change some of it. The particle number is also not constant, so it is highly. It looks to me very consistent with the idea that you're in the positive temperature part of the Onsager.
Right.
And that's the natural place to be. Yes, please.
Did you try to derive the point vortex model from the complex Ginzburg-Landau equation? Because you can do it by asymptotic analysis.
You can derive. I know you can derive it from what do you call it? Nonlinear Schrodinger equation, but I don't know that you can derive the point vortex model from complex Ginzburg-Landau. Can you?
I think you should be able to do. I can't give you a reference for where it's.
Okay, because we did try that actually. We looked into again we have this derive phase and amplitude equations from complex Ginzburg-Landau. Take the gradient of that, try and derive some sort of equivalent to an Euler equation and go from there. But we found there were extra terms that
You have to do it by making a model of the vortices from looking at matching, doing asymptotic matching, and then you can work out the interactions between them.
Okay, we can talk about this later. Yeah.
There's papers by people like Pisman and others that have done things like that.
Just now about the nonlinear Schrodinger equation, and this is quite similar to the nonlinear Schrodinger equation. No, and I know with that equation you have kind of stable in time soliton solutions. Are there any such solutions for this? And do they kind of give you stable
in time,
temporally stable points, or temporally stable vortex?
Are you talking about when you say this? Do you mean for the complex Ginzburg-Landau equation?
No, for the nonlinear Schrodinger, but it has the same form of potential.
Right.
So I just wondered if this also has, soliton solutions that would be like kind of.
I'm not sure off the top of my head. I think it does, but I'm not sure.
Okay, I think we are running out of time, so please thank Pearson again.
Beautiful pictures. I don't have a point to make. It's man-made locomoting means the biology. Okay. So this morning, at times I check Google images. Okay, this one is amazing maybe from some millions or billions of years, terabytes. A friend of mine just sent me that. They were doing something like that, and suddenly they were buried and preserved. So the Reynolds number is 10 to the minus 3, I guess, here. And here, you see this big blue whale. It's an artistic rendering, I suppose, for the size. So yeah, it's not real photo, but you have some photo similar to that. So if you calculate the Reynolds number, that's 10 to the 8, I guess, suppose, something like that. Anyway, so that's how I did my discussion. So do something, right? So here you don't have to have turbulence. The flow can be very coherent. Maybe this so-called inverted von Karman state is generated in order to produce the force, but it's not necessarily a turbulence at Reynolds number 10 to the 8. But here, when the density is high enough, even at the Reynolds number 10 to the minus 3, you can have turbulence features, like the topological turbulence we heard just now.
You take 1,000 blue whales and put them all together, they will interpolate.
You have to pack them, and they become very depressed. Yeah. Right. And they will be happy, or they will be forced to be washed ashore, and they're sentimental animals.
I do have a question.
Yes.
So maybe this is for Zonemir. For an active matter system, has there been a measurement of a cascade directly? Say from velocity data, PIV data, something like this? Like what Luca showed as a prediction from his active pneumatic model?
I know in this microtubule-based system, it has not, my student is doing it, but there has not been a measurement. But bacteria, maybe there has been, I'm not sure.
Has there been, I wonder if there's been one in a bacterial suspension.
I don't know.
There is the paper in 2000
I wouldn't be surprised.
with DNAS. It's just that the scales are too small, and why hasn't there been a direct
It could be a separation of scales issue. Yeah, so what's the difficulty?
That's a bit typical question.
I mean, what you find is that it's all controlled by the boundaries, right?
Yeah.
So really to get the bulk behavior, you need to compare, centimeter-sized fluids that, that last for a long time to proliferate, and you need to reach them. So you need to go to bulk materials that are active. I think otherwise you'll find out that you're answering against the boundary.
No, but I mean, just following up, in what Jon Dunkel and company did, they have basically sort of quasi-2D system confined between plates, and I think there they do have velocity statistics and cascade and so forth in that context. Rather than viewing it as dependent on the boundaries, you view it as a system without momentum conservation, but a 2D incompressible velocity field and activity, and ask what you get there, and you get something. Now, it's very different from ordinary turbulence because you're completely Stokesian, and it's really the polar order parameter that's the driver rather than the velocity field. So there you have something.
Yeah. Other comments?
Yeah, maybe I can make a comment about that.
Luca.
And I will actually use the microphone.
Since I still have it. So I think we need to clarify what we mean by measuring a cascade or measuring the spectra, because one thing that you realize is that the two things are not exactly equivalent. So, for instance, in all this data that I showed you, there is clearly like a spectrum. However, this work by Jean-François Joanny, Casademunde, and Ricardo Alert, they even managed to measure to sort of address directly the cascade to see if there is a flux that goes in whatever direction. And to my surprise, they showed that indeed the energy remains localized at the scale, at the active length scale, which is somehow the scale of injection. So they use a specific parameterization in which essentially the pneumatic texture has no defects. So that's a way to speed things up. And so to me, it's unclear whether this peculiar feature is actually creating this localization as an artifact or not. Because I naively I would have thought that when you have a complex fluid, somehow energy can actually be transferred from one field to another. I mean, there is not just the velocity field, right? So it can go from the structure to the velocity, perhaps back to the flow in some funny way. But I think this is very interesting, and so far it seems that there is a little bit of evidence that this is not happening. But I don't know how much unbiased is this approach. Please. Yeah.
So I know a little bit about this work, and as far as I understood from Ricard, there is actually an argument where this term that would be responsible for the cascade, the nonlinear term, is zero for the velocity. So
Can you use your seat mic, please?
So I'm not an expert on this, but from what I understand from their work, from what I heard, is that they actually can show that this nonlinear term that would be responsible for the cascade should be zero by symmetries. So like from theoretical arguments.
This is in
This is Ricardo Lerta, Joani, and
okay.
Yeah.
So
that's
I just wanted to know which model.
Yes. So I think it's not. I don't think it's related to the defects. I think there is a general argument that says that.
It is true, though, that you also use other So, for instance, I know that in their work, I mean, to essentially to speed things up as much as possible, they take the flow alignment parameter. Now we are digging into the technicalities. They take it equal to 0, and which I don't know what that implies.
I'm just saying that it's not a numerical fact.
Okay, sure. No. I wasn't thinking about the numerical artifact. I was thinking about a consequence of the choices that they make in the analysis.
If I can just ask a technical question, do they just have a co-rotational derivative?
Exactly.
Right. So the point here has to do with the form of the transport nonlinearity that sits in the
Exactly.
convection equation. So if you have a stress that's produced by something that looks rod-like, then you have an upper convective derivative. But if you're assuming that your microstructure, and so then you align with directions of extensional strain. On the other hand, if you're assuming essentially that you have spherical particles or round particles, then for the stress created by such things, then you just have a rotational derivative, and you don't have an alignment in the same way. And change the stability of systems quite a lot. So that's the point you're making. Yeah.
Sorry, Mike, when you say you don't have alignment, you're still thinking of systems that carry stresslets but structurally don't have alignment? So they have an axis defined by their force dipole, but
that's right. They're just rotated by the vorticity. That's all that happens to them. That's the form of the Jeffreys equation in that case. So I've always been somewhat confused by the symmetry theories, because when you do the coarse-grained microscopic theories, the geometry of the particle plays a very large role, and you don't have a free parameter, so to speak, unless you change the parameter or change the geometry change of the active particle. Then you change the transport derivative in a very deliberate fashion. Does that make sense to you?
So, sorry, just to see if a non-expert followed that, did you mean in the first case with the upper derivative that when the particles do have some preferred axis, that you get more potential for alignment, basically?
Yes, that's exactly it. So imagine you just take a rod-like thing that's in a straining flow. It will line up with the direction of maximal strain.
Right, but the spheres.
If I take a spherical particle, it will just sit there unless there's a background vortical flow that will make it rotate.
I have a question for our program for this conference. Do we have a talk on viscoelastic turbulence?
Yes, we do.
We have?
Sandra.
Okay.
Raise your hand.
Oh, okay.
Yes,
Today? Tomorrow?
On viscoelastic turbulence.
Friday morning. Okay. So we have very good coverage. Excellent. Reynolds number around small. Right. 10 to the minus 4. Okay. Even below these guys. Okay. Right. Yes.
Sorry, I have a question as an outsider.
when you talk about turbulence, I mean, in the conventional turbulence, let's say that there are many turbulent seas. The mechanism for the cascade is quite obvious. It's a nonlinear term. And my question is, in all of these beautiful models that you have presented, where are, in fact, the nonlinear terms that could replace the standard inertial term? Because, I mean, if the question is where does the cascade come from, you have to identify the mathematical terms which are responsible for that. The fact that Reynolds is very small doesn't mean that you don't have coherence, which is another property of turbulence, because, as you know, hydrodynamic correlations are very effective at almost zero Reynolds, which is very different. Very small is very different from zero. So my plain question is, where are the nonlinearities in these equations which might replace U grad U?
May I answer that?
Yeah.
Please.
So
Yes.
I will say something about that in my talk after lunch.
Okay.
So I won't do any spoiler right now.
But can you tell me now, since we are...
I'll tell you now, so you can decide.
Suspension.
No, I don't want to skip your
As far
talk.
as we can tell, at least in the regime we've looked at, the nonlinearities in the orientation in the vector order parameter equation.
I see.
The kind of self-advection of the polar order parameter is certainly very important.
I see.
In regime.
Okay.
And in fact, U dot grad U isn't important in that regime. The velocity fluctuations are.
No, I understand. So there is another mechanism, okay, for self-advection. That makes a lot of sense. Thank you.
Oliver?
In Luca's language, it was the advection nonlinearity in the Q tensor advection of the stress. Also has a convective nonlinearity.
And why would
Version gives you the
that be?
There's interesting differences between the polar and the apolar case.
Okay, but why would this term be significant while standard inertia would be silent? If it is advection.
because
it's a different advection velocity then?
Yeah, no, but in this case is the structure, is the configuration of the rematics that is actually advected. So it's not the velocity. Yeah. In fact, in all my simulation, I actually set the convective derivative of the velocity to 0, precisely because I want to achieve 0 Reynolds number.
I see.
That's what Luca was commenting on.
Again, I mean, so from this more recent, numerical data, it seems that there is not cascade. But I don't fully understand all the details. But one thing that we can propose, I mean, so they have a method actually to see how energy is localized across scales and how it fluxes. And so perhaps our friends experimentalists in the back of the room can actually use the same method on their experiments, and they will tell us. I mean, so far, one thing that I realize is that, to my great surprise, that these equations work extremely well. I mean, surprisingly well. And so I tend to trust them. And so, if the equation says something, probably also the experiment will say the same thing. But obviously, I mean, we need to check. Did you hear me, Zvonimir?
Yes.
Very good.
You wanna comment on that? No? Okay. The other experimentalist, William? William, you want to add something? No? Okay. Actually, I had a question for you for your spinning particles. What is the source of this effective surface tension? Just want to make sure. I heard, I was asking somebody else, and the source of effective surface tension in your
it's
system.
So two dipoles, if you average over a rotation, attract each other. So it's the magnetic attraction between the dipoles, and it's about the right order of magnitude. If you estimate the surface tension as an interaction energy per size of the colloid along the interface, you get about the right number.
Is the order of the surface tension of water?
No, it is significantly smaller.
Okay. I remember this magic number 72 for water, dyne per centimeter. Right.
But water is it's even more than kT per molecule.
Okay,
Right?
really?
And in our case, it's sort of maybe more than a kT, 10 or 50 kT, but per micron.
Hmm.
So that reduces it a lot.
And the kT is your random, your kinetic energy.
The magnetic interaction energy, the distance of about a particle, is of the order of between 50 and 100 kT.
No, you're saying kT being the surrounding temperature or the kinetic temperature?
Oh, no, it's just if you want to compare it to a colloidal system where diffusion is important, I'm putting it in units of kT, but kT is irrelevant for us.
Right, okay, that's what
Oh, please. Definitely.
Maybe I ask something very naive, but maybe could be interesting question. Because turbulence, if we reduce down to a system, usually should be described by a set of the Turbulence parameters. Like we talk about Reynolds number or not. But different systems have different like agitation, dissipation, interactions. Even be nice to have a little more systematic way to normalize such a way. It cracks down to a set of known or uniform difference parameters. Yesterday, I mean, for past 2 or 3 days, we heard quite a few. Reynolds number is always one, but they are very different. In reactive fluid like this, we have some like very easy to grab for people outside your field to understand why that effect can be or cannot be important, and so that's when look very fundamental, but could be very important.
So let me try to understand the question. You want to find a demarcation point that.
Well, some control parameter, which is dimensionless.
Okay.
If that take one limit, equal classical fluid mechanics, take another limit, I mean, making up example, become cosmology, for example.
Mm.
So that would be very unifying and satisfying. Can you see something like that, or I mean, not to you, to the audience? Since we are come for different kind of communities, and without that kind of simplified version, it's hard to communicate. Thinking,
Mike.
So in the literature of liquid crystals, there are these dimensionless number. I mean, a famous one is called the Ericsson number, and is the ratio between the elastic stresses essentially versus the viscous stresses, and tells you the importance of backflow. I mean, how much a distortion of the director can actually drive flow. But it's not crucial for this problem, I think. I think in this problem, somebody, now I don't remember who, so somehow used some active version of the Pleclet number, in which you have sort of like the velocity due to the activity on the numerator, and I believe the rotational diffusion on the denominator. And somehow this number tells you how much this active pumping of stress into the system is relevant compared to instead the tendency of a distortion to actually spread and becomes uniform across the system. So I think that could be one of these dimensionless number. Yeah.
I think that's similar to this. I think it's square root of k over alpha.
Yeah.
Actually, because
that's all it is.
Yeah. In the end, it is. Yeah, you're right.
It is like any Pleclet number, all the kinetic coefficients drop off. It actually ends up being the ratio of a length scale built from stress and elasticity only.
Self-organized criticality, my understanding is it doesn't have such a
scale.
Scale. It's scale free and everything parallel.
Right.
So that's another beauty that you don't have to have one if you can get away with that. But I don't know how much it can be applied. I mean, we're trying to hear. I didn't understand everything, but 14 maybe it could be self-organized critical. But is there anything here? Is like that nature that you don't have to have an independent parameter control? Okay.
Good question. I don't think these systems are running near the point of criticality of any sort. So one can think about something like that, but maybe unlikely for these systems, I would say. So we're hitting the point. Lunch?
We're at lunch.
Okay. Yep. Okay. Happy lunch. Yep.
Come back.
Okay.